Welcome back to Microbiology. I'm Lauren Yost, and I'll be your host for the evening while we talk about microbiology and basically what it is. So that's really all that this chapter is going to be about. Yes. So this is not going to be a little interesting, fun little thing. Just going to give you a little bit of a heads up for what to expect for what's coming up. We're just going to talk about the purpose of the course essentially and the materials that are needed. And then we're going to get into the microbiology stuff, talking about cell theory and germ theory and the contributors and contributions to microbiology as a whole, how to write a scientific name, and the tree of life. So first off, talking about the syllabus, just a quick little rundown for it. The syllabus is where you are going to find the lecture and lab schedules. You can find all of this on Canvas as well, just so you know. And that's where you're going to find all of the communication with me. Canvas is the place to go as far as any announcements that I had to put up or, date changes and assignments. That's pretty much going to happen there because nobody ever goes to lecture. So if you are somebody who is, in the in-person class. And obviously, if you guys are in my online course, then you're not going to go to lectures. This is as good as it gets. So please do keep up to date on Canvas. That's where you're going to find any documents that you need to print out for the lab, any homework that you have to do, or any of your grades will all be posted there. And I'll try to keep up with them. Just keep a lookout for those zeros that come out of nowhere. All right. What homework do you have? We have chapter homework and we have lab homework. They're both on Connect with McGraw-Hill. It's a link on the side, on the left side for Canvas. So just, be aware of that. That's where you would find the McGraw-Hill link. But the assignments themselves, you can link to them through the actual assignment section on Canvas on the left side where it says assignments. So you can get to them that way. And there are also links through the lessons in the modules as well. So just be aware of that. Yeah, and no time limit on those. Yeah, they're going to be like it says on there, mastery essentially. So you can do it as many times as you need to get 100% while it's open. It doesn't stay open the whole semester. It has a due date. So projects, what projects are you going to have? There's two major projects. The lab unknown project, we give you a mixture of bacteria. You split them apart and figure out what they are using the tests that we provide or that you learned about. That's the lab unknown project. And then we have the case study presentation. And yeah, it's what it sounds like. It's a presentation where you're going to be up in front of people. If you're in the live person class, that you will have to really get up in front of people and give a talk that's expected of you as part of your grade. You automatically get 50% off of your grade if you do not participate in that part. Yeah, online people, just you'll be submitting videos, and I will get into a little bit of the details for that when we get a little bit closer. That'll be more like unit 3. So, all right. The lab stuff, wear a lab coat. Have to wear a lab coat. You have to wear closed-toed shoes for protecting of your feet, right? And if you want to wear gloves, you're welcome to bring them. We will not be providing gloves. I mean, most of the stuff that we're using in our lab is not particularly dangerous. And as long as you know how to wash your hands properly, which is part of what you learn in this class, learn how to wash your hands properly and do that and keep everything disinfected in the room, and it should be fine. So that's the idea there. And of course, you wear a mask. You're always welcome to wear a mask. Absences, you can't get more than 6 absences. So you can't get 6 absences or more than that is what I'm trying to say. They're maximum at that point. And once you hit 6, we will fail you out of the course. Don't freak out too much about it. Essentially, you have the opportunity to make things up if you have legitimate reasons for having missed the course. If you just missed it because you slept in or you missed it for whatever, you're either going to have to take the hit of that absence or hope that you don't, basically hope that you don't get to the 6. It didn't happen in my class, and it didn't happen in Dr. B's class that I know of. It's never happened to either of us ever, ever. So it shouldn't be an issue. We put the bar kind of high for that. And if you have a reason you come in with a doctor's note because you missed because you had COVID or something, and I still have the resources available to make up that lab, then I will, discount that absence. But if you can't make it up, then that you didn't do the work associated with that lab, and you missed it. So I can't give you credit for having participated in. That's what that's about. All right. Pre-lab quizzes every single day before, at the very beginning of lab, the first 5 to 10 minutes of lab, we will take a quiz using our clickers. The clickers look like this. And you will have to get these registered on the Turning Point website. If you haven't, it's on the left side of your Canvas over there. You have to associate the number on the back here with your actual name, like your license that you have purchased to go along with your clicker. So if you need help with that for whatever reason, you can contact me, but I feel like the instructions should at this point be pretty straightforward. Buy your clicker, buy your license. The license is cheaper if you buy it through the Canvas thing, through actual Turning Point link on Canvas. And then register your clicker with, according to what your name is, so that when it pops up, I can actually give you credit instead of, CX5432 credit, but I'm not going to, look that up every time. I'm sorry, that's not happening. So that's the idea with that, especially when I have 5 sections that I'm teaching. All right, moving on. So that's the idea with the clickers. We will do the pre-lab quizzes before the lab with those clickers. We will also use the clickers for our exams in our lecture and the exams for the final exam. All of that stuff, the exams for the labs too, will also be using the clicker. So we'll be using a lot if you are in the in-person class. If you're online, which most of you watching this will be the online type people, if you're watching this and you are an online person, obviously your exams are going to be on Respondus, and your final will be on ProctorU. So that's what you'll be dealing with there. Special circumstances for those of you online who are just not having it with Respondus or ProctorU can contact me, and we'll see if we can figure something out. I can't put everybody in the testing center. So, but contact me and we will see what we can do. Not everybody. So I'm just saying that. Of course, that's the benefit of those, is that you can take the test really late at night. So you can't do that really with the testing center, of course. And like, even if you take the tests with me in my classes or off hours when I'm not normally at work, I sometimes meet people for makeup stuff. Even then, I can't see everybody, and I'm not coming up here at midnight. So I'm sorry, but I'm not getting paid that much. So, but yeah, so that's the idea. So I work with people. So if you need me to work with you about something, don't take advantage, please. But, if you need me to work with you, I'll work with you. Moving on, grades. The breakdown of the grades and like what percentage is what in your grade scale is going to be available on that syllabus as well, which of course I didn't pull it up before I gave this lecture, but it is available on Canvas. The schedule is there, as well as the syllabus that has the schedule on it, and the grade breakdowns as well. So all of that should be on Canvas: schedule, grades, all of it. I'll try to run down a little bit if I can remember. I believe the case study presentation is 10% of your grade. The unknown lab project is 5% of your grade. The chapter homework and the lab homework each are 10% of your grade. The pre-lab quizzes are 5% of your grade. We will drop the lowest score for that, and we will drop the lowest score, I believe, for the lab homework. I'm sorry, my throat today. What else? Exams, right? So each unit exam is 5% of your grade for each. There are 4 of them. So that's 20% of your grade overall for all 4. And then another 20% for the final, in the lecture final. And then we have the lab stuff. The lab midterm is 7% of your grade, and the lab final is 13% of your grade, for a total of 20% of those exams. I think that's everything. You can get up to 3% extra credit, and those will just be random You never know. A lot of them, for the in-person stuff, a lot of that is going to be associated with, I can't remember what I was even going to say. It just like emptied out of my head. A lot of that's going to be associated with, I don't even remember. I don't even remember what I was going to say. Anyways, it's mostly geared towards, sorry, it's mostly geared towards, discussion type stuff. For the online people, discussions on like posts that people make, or like, whenever you post your, because you guys have to post videos for your presentations. So whenever you guys are going to be having comments on there, there will be quite a lot of opportunity made up there for the extra credit. Whereas the in-class people will have that like randomly interspersed as questions throughout the lecture. I guess I'm getting to that. Okay, moving on. Disability, if you guys have any disabilities or think you might have a disability or have a diagnosed disability from an actual doctor, please contact Terwana or TJ Brooks. She is at this number listed on here on this slide, and that's a good person to talk to if you think you need accommodations for whatever your situation may be. And then she will email me, and then I will follow whatever those rules are that are presented to me. And then all due dates for all of this will also be on the last page of that syllabus. So you guys will have those available. Of course, that can change, and I would update you guys on Canvas about that if it did. And I'm done. All right, moving on. You need the textbook and you need the lab manual. Okay. You need a lab coat. You might want this. So this is a textbook versus so this is for the chapter homework is this, and then this is the lab homework here. Right, and then this is the lab manual, and then these are the clickers. You saw mine was the one here on the end here. I have a few of them. There, I keep them as extra because people do forget them in their car or whatever, and I don't mind loaning one every now and then, but that I'm not giving any to anybody permanently, so that ain't happening. They're mine. So lab coat required, closed-toed shoes required. So this is where those of you who are in lecture would be participating in this portion of the question asking process, and you could potentially get extra credit for this. For those of you who are on online, you're gonna have to sit through these a little bit real quick, just so that you can get an idea of how it might work. So essentially, like if this was a question that was coming up, you would on your clicker want to hit A, and you would just hit. Let me go ahead and show you what it would look like. It's not going to contact it. Plug this in. Maybe it'll just let me. Okay. Anyways, so then you would put in A, and then you could hit the little enter button in the center, which is that line that's all the way across, which is this one right here. I don't know why this isn't linking up. Anyways, and then you would hit enter on that. So A, whatever, man. I don't know why it's not connecting. And then go on, and then it would like let me give you everybody a score and tell you what the right answer is. So I obviously you guys can't answer online, but this is meant for the in-person lectures. But if you guys are willing to discuss this, there is a discussion post on Canvas that allows you guys to like introduce yourself and say your favorite food or whatever, and go meet each other, get to know each other. That's the thing that's lacking the most for the online course, I think, is that interpersonal relation. It really lacks there. And I think it's a huge detriment to the learning process, is that you guys aren't like communicating with one another. So you just think you can do it all on your own. And this is actually a pretty difficult course to do kind of on your own. It works well with partners. So moving on, prokaryotic organisms. This is a question where we would have more than one choice. So in that case, you would literally just type in A, and B or whatever. So prokaryotic organisms would be bacteria and archaea. So you would put C, just put a C and an E, and then put the enter button. And then that would be the answer. Here we go, C and E. And this is one where it would be a ranking. So it's like I would tell you like from 1 to 10, how confident do you feel about using a microscope? Or in this case, how many hours a week do you think you should spend studying microbiology? I'm not going to tell you how many hours a week you should spend studying, because that differs so much from person to person. Some of you need to study very little. You just read through it and it like sticks in there somehow. And then the rest of you need to put a lot more effort in. Do what you need to do, but do be aware of it. Like if you were to say you think you should study in a week, maybe, I don't know, 7 hours, like an hour a day at least. I think should be at least an hour a day. Personally, I think it should be more than that, probably 2 hours a day, especially for a course like this, which is so new to you guys. But you do what you need to do, man. But if you think it's going to be 2 hours a day, then set aside 2 hours a day every day and commit to it. And you will do a lot better if you do that. All right, chapter 1, let's move on. Yeah, not just talking about the syllabus. So microbiology, a microorganism, that's a term, and this will absolutely, almost certainly, I haven't written the test yet. Of course, this is just chapter 1. I haven't gotten to like you guys' specifics yet, but this is a question that has appeared on my tests before, at least twice. So be aware of the term microorganism. What is a microorganism? It's any organism that is too small to be seen with the naked eye. You need a microscope to see it. That's what that means. So we're talking about anything that you might call germs or bugs, or agents and viruses. A virus is a very specific thing, and we're going to get into what a virus is soon. It's not bacteria. All right. Microbiology includes, when we break it down to different factions or whatever you want to call it. I don't know. That sounds so like, what was that movie with that girl who cut her hair? I can't remember. Shailene Woodley or something. And I read the book and I feel bad. It had the factions. Anyways, study of bacteria. If you're studying bacteria, that's bacteriology. Studying fungi, that is mycology. Worms, that's helminthology, because they're helminths. I know we had to make it a weird word and couldn't just make it wormology or something. Studying the immune response is immunology, and then studying viruses, which are a different thing from all of this, believe it or not. That's virology. Okay, so if you studied HIV, what sort of microbiologist would you be considered? You would be a virologist, right? Because HIV is human immunodeficiency virus. All right. What do microbiologists study about these things in general? Well, they're going to study cell structure and function of whatever organism they are studying, growth and physiology of that organism, the genetics of that organism, and they might say genetics of that organism compared to the other organisms in whatever faction they were studying, taxonomy and evolutionary history, and then interactions with the living and the non-living environment. Sorry, I have an appointment in like 30 minutes. So how does it interact with the things around it? That's what it's talking about too. So then we can break it down even further into other types of microbiology, not the factions so much. You might even have factions falling into like medical virology or something like that. So now we're talking about what kind of microbiology. Medical, obviously, medicine, like diseases. Public health microbiology and epidemiology is a little bit different thing, right? If we're talking about like the incidence of a disease all around the world or something like that, that's what we're getting into with this, right? And like the frequency and the likelihood of spreading it or anything. All right, then immunology. This, like we said, the study of the immune system, basically how our bodies, typically people bodies, are going to fight off disease. Industrial microbiology, how we can use microbes for our like purpose of like, whether it's breaking down oil or making fuel or something like that. Agricultural microbiology, that would include the microbiology of the like the soil and stuff, and how that affects the growth of the food sources that we eat. That's a huge issue actually right now, as far as like nitrogenization, nitrification, and stuff like that of the roots of plants. So getting the nitrogen into the soil, that's been a big issue lately with like global warming and stuff like that, of course. So moving on, environmental microbiology, how like these, like we see a red tide, which is like this red algae stuff growing up over here in this picture in the lower left, and then the dead fish as a result. Why is the algae growing up, overgrowing like that, and then causing them to produce toxins or blocking the oxygen in the water for the fish so that they can't get air? Essentially, they suffocate. Okay. That's the environment. These are agencies that might have to do with things with the microbiology. A lot of them focus more on the disease sides of things, but not entirely. We've got agricultural in there as well as food microbiology, which is something that I used to focus on. CDC, Centers for Disease Control, the USDA, WHO, FDA, all of these, and the NIH, I cannot speak today. The NIH and I don't know OIE. I'm sorry, I don't. Okay, microbes. All right, so what is cell theory? Cell theory is an important concept in all biology, not just microbiology. Cell theory states that all living things, if it's alive, it's made of cells, at least one cell. And we will talk about what constitutes a cell. Then all cells come from pre-existing cells. They can't just spontaneously generate. All cells have DNA, which is the genetic material in the cells, and all cells contain a membrane. So that means they're covered by a membrane always. Now, when we look at this picture down here, these are examples of cells. We have eukaryotes and bacteria and archaea, which are prokaryotes, which we'll talk about the difference in a moment. They have a membrane on the outside. They have DNA on the inside, and they come from other cells. But viruses don't have that stuff. They can have a membrane. They don't have it from themselves though. They steal it from the host cell that they were infecting. They're tiny. They live inside of cells. They bud out of the cells, like literally what it sounds like, little like push through, and then it encloses around their little insides. And then that it creates the envelope, but that's not their own membrane. They stole that. And they have their genetic material inside doesn't have to be DNA. It can be DNA or it can be RNA, which is like, we use that as like an intermediary, a sort of blueprint. You have DNA of the whole genome. RNA might be made of a gene for a specific protein. The RNA encodes just that. Then they use that RNA, like that copy of the blueprint of just that one part of like the house. Like this is the bedroom part. And then we make proteins out of that. So it's DNA, RNA, protein. Viruses don't follow any of those rules. So they do their kind of their own thing, and they are not considered living. Okay. They definitely don't have cells, and they are not considered living. So which of these are acellular, meaning they are not cellular microbes? And that would be viruses, still considered a microbe, but not alive. All right. Where can we find the microbes? Everywhere. Not on the moon. There's no life on the moon, please. And then everywhere, polar ice caps, Earth's crust, oceans, inside of plants, inside of animals, all over everywhere on the Earth, pretty much. If it's there, it's there. It's way down deep under. You can go down 7 miles in the sea under the Mariana Trench, and then you can dig up the soil that's underneath there, all that mud and stuff, and there's even microbes growing in there. Nuts. So, we see them all the time in the news. If you guys see any stuff about it that you find interesting, please start a discussion in the Canvas thing. I'm pointing off to my left, but I feel like, is this accurate what you're watching? I don't even know. I can't tell. Yes, that seems like it would be accurate. I don't know. Anyways, yeah, you guys know. Start the discussion. And maybe or not, there could be occasionally extra credit involved and stuff like that. So, get creative, get interested. And then, in the environment, and we talk about evolution. When I talk about the evolution of microbes, it's indisputable. It's indisputable that microbes undergo evolution. How can you say that? Because we used to just have E. coli and staph and gonorrhea or whatever. And now they're all getting drug resistant. That's evolution. Just saying. You can take a plain old E. coli. No, I almost said a bad word again. No joking. And you can take plain old E. coli, just normal old E. coli, put it on this agar sheet and have it grow. And it'll be a no antibiotics and just go increasingly stronger antibiotic. And at first it'll grow up to that line where the antibiotic appears and it won't go anywhere. But eventually there are outliers that have mutated and they'll grow and they'll reproduce and then they'll keep growing all the way across. That is evolution in action. You can see it happening. They did not have any genes at all to protect them from that antibiotic. That just comes out of just mutations on their own. So, and then of course we can get the development of new strains as well. Good, we're good. Okay. Disease. What does disease mean? It's a term. It doesn't we think we know what a disease might be, when you have a disease. But what does it really mean if you had to define it? It means it's anytime that the body is different from what is considered normal would be a disease. Now, that can be caused by microbes or it can be caused by other things like cancer or whatever. So we're mostly going to be focusing, of course, in this class about the microbial causes. So think about it that way. Germ theory of disease. Some diseases are caused by microbes. So we talked about that, right? We just said it. We just said it. So obviously some diseases are caused by microbes. Germ theory says that if you have a disease and you can prove that it's caused by this organism, then you can break it. It's called KOCH postulates, and we'll talk about them soon. But that's the idea that some of these diseases, they're not coming out of the thin air. Like they used to think the Black Plague was caused by miasma, which was just the bad smells of the people around and the dead bodies and whatever. So they would wear these long masks on their face, almost a beak on them, and they'd fill them with flowers and stuff like that to keep out the bad smell because they thought that was keeping out the sickness, because the sickness obviously was the smell. Incorrect. Those are plague doctor masks, by the way. Incorrect. No, that was called miasma, and that is incorrect. Disease is caused by germs, by microbes. So a pathogen is the term that we use for any agent that is, a microbe, viruses, bacteria, fungi, protozoa, helminths that cause disease. They essentially act as parasites within our body. They live within us, causing us harm, so they can kind of benefit from that, right? That's a parasite. Just think of ticks do that too, right? That's a parasite. We would consider that a parasite. Okay, lots of microbes out there can cause diseases. I got this, causes of death. These are diseases. Clearly not all of these are microbial, and that's pretty much what I'm trying to show you here. The leaders are always going to be cardiovascular diseases and cancers, pretty much across the entire world. So you get to respiratory diseases, and that's not just talking about the ones that are caused by microbes. Okay. Vaccination. What in the world is that? What is the point of it? Why should we be worried about it? Vaccination prevents disease, and we will be talking about how that is, and it is my favorite. So please be excited, or at least pretend to be excited, because it's my favorite and I love it. So you should so get excited about it. The immune system is an amazing thing. It's so crazy that we only have 20,000 genes in the human body, and our body can respond to over 8 billion different kinds of bad things out there. Isn't that crazy? That how you would think you need a gene for each, but you don't. Our body has found a way around that. So that's what the immune system is about. And I can't wait to share that with you later on in Unit 3. I hate that I had to wait that long. All right. And then we'll talk a little bit about skepticism and the anti-vax movement. I don't want to get too much into it. When Dr. B wrote some of these slides, I had to go back and edit them. There's a lot of this. He's real angry about that. So of course I am going to bring up the benefits of vaccines. I am an avid, proponent of getting vaccinated. Help strengthen your immune system. Why not use this tool as your, advantage? We've eliminated smallpox from the whole planet with vaccination. We're almost there with polio. Afghanistan and Pakistan are fighting on that one. They still got people coming out sick with that, but still, it's pretty crazy, right? So this is where we were before. If you had this many, this is what vaccines have done for us, the cases that we see now. So this is in the US. So pretty cool stuff. Definitely vaccines work. I'm not going to argue with you about that one. I'll probably just kick you. And then they definitely work. As far as like the bad side effects, we can discuss that. We'll discuss some of it. Of course, there's going to be some bad side effects because when you're trying to turn on somebody's immune system to fight back against something that's bad and your normal response would be bad, it's going to be bad to the vaccine sometimes too. It has to be. That's the body building up the immune response. Anytime that you are sick, you go get sick, you get sick from the cold, sick from the flu and whatever, and you feel that pain and that ache and that just ickiness and that tiredness, that soreness and the swelling, that's all your immune system. 100% your immune system. The virus, the bacteria is not doing that. Your body is doing that. That's what your body is doing to fight the thing. So sometimes you need to kick that into gear, and you'll feel that too when you get your vaccine. And I have to say that just because people think it should be just nothing, no response. But yeah, your body's going to react to it. Should. That's a sign that it is reacting to the vaccine and responding the way that it's supposed to. So that later on in life, when you get re-exposed to the thing, the actual thing, there'll be no symptoms at all. You just won't even know you were ever exposed because you already had it kicked into gear. So that's the goal with vaccination. Measles, I don't really want to talk about it. It's coming back up because of anti-vaxxers. I don't want to talk about it. All right. Humans, how do we use microbes? Yeast give us beer, wine, and bread, and fungi give us cheese. Moldy bread was used in Egypt to treat wounds. Cool stuff, right? Because penicillin, we discovered, comes from mold. They figured it kind of out in ancient Egypt. Good. Okay, we're doing great. All right. History of microbiology. So I guess not microbiology, of microbes in general, like the evolution over time, right? Where did they come from? When did we see them on the planet? There's some discussion about where they may have come from originally. Did they actually generate here on the planet, or were they introduced by, maybe there's this theory called panspermia, which I'm not going to get into too much, but it's the idea that like an aspect of a microbe or some parts of a microbe came in on like asteroids and were introduced to our planet like way a long time ago. That's not meaning to say that like that's aliens coming or something like that. It's just like they were already on it and happened to land here. So I don't know. Those are theories, right? We can't go back in time and really prove any of that, unfortunately, not right now anyways. So we're just going to move on. I'm not going to say where they came from originally, but it seems to be that they probably came around onto Earth over 4 billion years ago, sometime around then. We know that these guys in this picture, these ugly stacks of junk. Yep, I have words that sometimes come out of my mouth that were about to come out of my mouth just then. These are called stromatolites, and they're like, they say a biofilm of bacteria. That's just like when bacteria kind of just keep growing up on top of each other and they kind of live together in like a community, right? That's what they do on your teeth too. It's called plaque. So, but this is a bigger version of that, and they have been seen to probably have existed up to 3 billion years ago at least. Really cool stuff. Yeah, they've been around forever, and we think that they came first. And then those were like bacteria and archaea. They do not have a true nucleus. They're called prokaryotes, pre-nucleus. Eukaryotes have a true nucleus. That's eukaryotes. U is true nucleus, which we will be talking about more later, of course. And then, yeah, so we think that we are eukaryotes, came from bacteria or archaea, or similar things that we might not know about from back in the day, that like one lived inside of the other and provided energy to it. And they became the things inside of our cells that like provide us energy now, that have their own DNA and stuff. Isn't that cool? Okay, the scientific method, we've all heard of it. What exactly is it and where did it come from? I won't make you guys memorize the steps or anything. No worries there. During the 1600s, we had scientific thinking developing. We figured out, we would formulate a hypothesis to explain a thing. And then we would try to prove or disprove it with like experiments or going out and observing or whatever, right? So it would idea first and then go figure out if it's true. So that's the deductive approach to applying this scientific method. That is testing a theory. So you come up with theory and now you're going to go back and test it to confirm it. There's another version called inductive, which I'm not going to ask you about, but just so you know what it is, that's observation leading to a theory. That's like what Sherlock Holmes does. Okay, so he's just kind of going along with the things and making all these observations of the little things and then getting to the answer there. That helped me understand what deductive reasoning was. So, what do microbes do naturally when they're just hanging out? So they can lead to food spoilage. We know this. We've had milk go bad. We've had bread go moldy, all of that. We've heard of salmonella and E. coli poisoning. That's what I used to test for, or listeriosis. And we talk about the environment. Obviously, that can affect whatever habitat that they might live in. Turns out 70%, oh man, 70% of photosynthesis in the whole planet is performed by microbes. That means they're the ones making our environment. So that is, sorry, there's some sort of bumping sound. Anyways, so that's pretty cool information that they're making our, atmosphere and whatever. And then we have symbiotic relationships with plants and animals. So that's another thing to, so it's pretty cool. I mean, they live in our guts. That's what I'm talking about, right? So they do some benefits for plants and animals and things like that. They're also involved in decomposition. They help break things down. Bacteria and fungi both do that, breaking dead things down, and they will recycle nutrients in the environment. Just getting into Louis Pasteur and Robert Koch. Only got 10 slides left. Louis Pasteur came up with that idea of that spontaneous generation was not a real thing. So spontaneous generation said that like they had a piece of meat out there and then it would have maggots on it. And then people thought that the maggots just came out because there was meat out there. No, he proved that it was flies that put them there. Yeah. So he did that. He came up with the idea of pasteurization, obviously, and the germ theory of disease. You got a disease must be caused by a germ. Robert Koch came up with Koch's postulates to verify the germ theory. He basically said that if you have a sick thing, it's dead, and you pick out a thing, you grow some bacteria out of it, or whatever it is, you grow a microbe out of it, and you're able to isolate it and then infect a uninfected organism host, and then it makes them sick with the same thing, and you can re-isolate it again. That's the causative agent, y'all. That's how it works. So that's Koch's postulates. That's how he outlined how diseases were caused by pathogens. All right, taxonomy. Moving on is the science of classifying a living thing. Carl von Linné, he was involved in classifying. He was the one that was involved in classifying. I mean, who cares what these people's names are really that much? Nomenclature, we're talking about species, genus and species. It's the binomial naming of an organism, right? So we're talking about, it's a Latin-based system where we have italics, and look, capital G here, little s there. Always has to be this. Always, always, always. When you guys get these questions on the test about it, this is how it has to be written, okay? Big first, little second, italicized, always, okay? So what's a species? Well, it's really hard to define what a species is, really, honestly, but we usually classify them based on like their genetics and things like that. And then microorganisms will push our limits of our ability to create schemes to organize and classify whatever group they might belong in. And so we're constantly changing that in response to it. Usually that's just based on whether it's the actual, like you look at their genetics and they seem so similar, but they're just acting so different. You might want to separate them because of that, but their genetics are so similar, you know what I mean? Like, it's just like, it gets complicated is my point. So give me just a second. Sorry about that. So here's some examples of naming common names like you see here, Staph aureus. So Staphylococcus aureus. So staphylol means bunches of grapes. So we see them all, bunched up here. And then berry, because that's the coccus, that's the round sphere shape. And you guys are going to have to know this stuff. You don't have to know it right this second. You will be asked about this on the lab exam, 100%, 100%. And then aureus in this case would be a golden. Okay. We'll go over more of that stuff later. And we see here Campylobacter jejuni is named for campylose is curved, bacterion is little rod, and jejunum is the part of the intestine that it was, mostly found in. It causes diarrhea. Then we have Giardia lamblia also causes diarrhea. A lot of them do, actually. And this was discovered. So we have names here. So we have Giard, which is the one of the a French microbiologist, and then we have Lamble, which is a Bohemian physician. So together, Giardia lamblia. So we have other weirder names here. We have chaos, which is named by Linnaeus, is a protozoan. Then we have Ahaha, the Australian wasp. Anyways, you guys can look through these silly little names, but just how people have decided to name them either after people or after words for things, or just silly stuff like abracadabra, abracadabra. These are my kind of people, guys. Okay, so if you were going to choose which one of these was correctly written, or why would you choose it, and how would you choose it? Well, we have Escherichia coli. That's E. coli. It's normal E. coli. And we would, like we said, we want to italicize it, right? And then the first gets the big letter capitalized. The second gets the small letter. So that's going to be this, and it's italicized. So that's the one that we want. Correct? Yes. All right. Moving on. This is back on that idea of eukaryotes versus prokaryotes and how we're going to classify things. We usually start off here. Eukaryote, U means true. Carry refers to the nucleus. True nucleus. If it has a true nucleus, it is a membrane-bound nucleus. Okay, it's separated from whatever else is inside the rest of the cell. So cells with a true nucleus, with a membrane-bound true nucleus, are classified as eukaryotes. And then we have the prokaryotes, the pre-nucleus organisms. That includes bacteria and archaea. They do not have a true nucleus. So they are classified as before nucleus. They exist before nucleuses did. So we don't usually use them to classify them as much, but we do use them as descriptors of the actual cells. How else would we classify things? Based on similar characteristics, and then based on genetics, typically. So we have phylogenetics is something that we use using the genetic makeup of an organism and its similarity, often like of the ribosomal subunits, which we will introduce those as well, and grouping things that way based on how related they are there, literally like the percent similarities. So we'll group similar organisms based on, and then put them based on their genetics, as well as if they have common ancestors from what we can tell, usually genetically, okay? These are the levels of classification. Do be familiar with these. I know you hate it, but this is the thing you need to know. This is the domain and the kingdom. They're often very similar in these microbes. So just FYI, if you were to look up a microbe and see what it was, they often have the same domain and kingdom, okay? But just know it's domain, kingdom, phylum, class, order, family, genus, species. I'm not going to ask division. And then I'm not going to ask about the strains and genotypes and stuff, but they would fall under the species if you're interested. So when we do talk about that stuff, now you know where it all falls. Know this junk. It's not like it's like that useful of stuff, but it is practical as far as biology goes. Like find out a little moniker that works for you guys. And I don't know, look it up or share it, whatever. And then we have new views of phylogeny and how things have like changed over time. Like I said, nowadays we often use 16S ribosomal RNA. That's the RNA that makes up ribosomes. And what are ribosomes? They take what I said of that. You take a gene in the DNA, you make it into like a usable little piece of that DNA of that gene that codes for that one thing. Take that part out from the whole in the form of RNA. It's a messenger RNA. Then the ribosomes come along and read it, essentially. So that's what ribosomes are. They're also made of RNA and proteins. But anyways, yeah, so we're going to be using that. Everything that's alive has RNA and has ribosomes. So that's why we use those, because everything has it. Everything, except for the viruses aren't alive. They don't have, okay? So yeah, we have the domains, archaea, bacteria, and eukarya. So that's the domains. Don't worry too much about that stuff. Yeah, and then we have the kingdoms of like, protozoa, fungi, animals, plants within the eukaryotes. So it's a separate thing. Okay. So your domains, archaea, bacteria, eukarya, and then eukarya will break up into, kingdom animalia, kingdom fungi, kingdom protozoa, and whatever. I don't need you to know all of those breakdowns. I just want you to know the list of like the bigger to the smaller. Okay, domain is the biggest, kingdom is the next, so on. Is this doing a thing? I don't even know. Okay. All right. So in summary for chapter 1, I want you to read the syllabus. Yeah, there's a quiz on Canvas about it. So you have to actually do it. And then if you do fill out the quiz, it's like, I can say, well, you said you read it. I get to do that thing that professors like to do so much. And then, what we talked about for the chapter itself, microbiology, it's multidisciplinary, study of microorganisms. What are microorganisms? You have to view them under a microscope, right? Can't see them with the naked eye. Microbes are everywhere in almost everything. Microbes again already, scientific names. And then remember, capitalize the first, lowercase the second, italicize, right? And then the 3 domains of life, bacteria, archaea, and eukarya. That's it. That's chapter 1. And I'm excited about chapter 2 because I love chemistry and I know you guys want to be reminded about it. I know you guys loved it when you took it. So I hope that I can help, refresh that love that you had for it so much. And I will see you guys next time after I take an hour to click out of this.